thanks for the kind invitation. And I wish I could be there in Lisbon instead of here in rainy Annecy with the ADVAC course, but be as it may. I'm asking whether perfect is an enemy of good for the reason that I've been doing clinical randomized controlled trials for decades. And sometimes I'm wondering if we're making things too complicated with our rules and regulations, intending the good, but maybe losing the baby in the bathwater. So I chose this paper, which is from 1987, and the first author is Juhani Eskola, who many of you know, and he was then a student of Professor Helen Mäkelä, who most of you have met, and she no longer is with us, but her spirit really is with us. And I chose this paper because she was always telling us that keep on thinking the KISS principle, meaning keep it simple, short, and some people say keep it simple, stupid. So they, after the meningococcal epidemic in Finland in 1970s, they launched a randomized trial on Haemophilus influenzae type B conjugate vaccine after realizing that the capsular polysaccharide vaccine was efficacious among children above 18 months of age, but not among infants. And it was clear that the conjugation to the carrier protein increased immunogenicity among infants, and therefore there were high hopes that it would also be efficacious among this age group. But the problem was on how to best demonstrate vaccine effectiveness of such a rare disease. Expectation in Finland was that we would have 120 cases in a birth cohort of 60,000. And also when exploring on what the ethical principles were in those times, it became clear that ethical approval would be possible for a close to real world setting approach in our country. So the study was designed in the way that it would be a nationwide open randomized controlled trial. Randomization was 1-to-1, meaning that those born on odd days, they would get the conjugated vaccine, and those born on even days would not get the vaccine right on, but only later on. And the endpoints was a hip cultured in the blood or central spinal fluid or other sterile fluid. All the central hospitals and labs in Finland were upgraded to detect culture-positive disease. That wasn't necessarily practiced at that time in all the remote hospitals. Then it was agreed that all the isolates would be sent to our lab in the National Reference Center in Helsinki. Also, the intention was to have a nested immunogenicity study so that one could measure the anti-hip antibody response and hopefully establish the correlate of protection. So you can see here the schedule on how the children that were born on odd days, those received the DTP and PRPD vaccine on 3, 4, and 6 months, and then the booster dose on the 14th month. And then those born on even days only received the DPT and then the PRPD once they were 2 years of age. And those who refused received no experimental vaccine. And please note that the consent was oral, and the nurses who were recruiting these children from their everyday practice in the regular health centers and well baby clinics, they noted down in the patient records that this family consents to this study. So within 5 months of the study, there were 14 cases of meningitis or pneumonia or arthritis or cellulitis. And you can see that 2 of those cases were among the PRP-DT vaccinated kids, and the rest of the 12 cases were among the non-vaccinated children. So very clear difference. The result when we calculated that into the vaccine efficacy figures was 83%, which was a very good result in comparison to the PRP only, which was 0% in the earlier studies. And the data on the incidence of Haemophilus influenza disease was, through the surveillance system, was instituted specifically for this study. But actually, that surveillance system remained and became the basis of our infectious disease register surveillance network. And in that way, this intervention trial actually did a tremendous capacity building on setting up the surveillance of invasive diseases in Finland. The future implications written in that paper were several-fold. It was clear that the results may also have implications for future vaccine development, in addition to the important clinical goal of preventing Haemophilus influenza disease. So the demonstration of that protective efficacy of the conjugate vaccine should encourage efforts to improve vaccines against other bacteria with polysaccharide capsules, meaning pneumococci and meningococci. And as many of you know, after these trials, the vaccine developers were quick on coming up with pneumococcal conjugate vaccine constructs, as well as meningococcal conjugate vaccine constructs, which then were tried out in several phase 1, 2, and 3 trials. So my lesson from this one is that, yes, I agree with Helen Mäkelä and Juhani Eskola that keep it simple, but think big at the same time. With this study, we were able to demonstrate the vaccine efficacy. We were able to establish the serological correlates. We were able to set up the nationwide surveillance system, which is the basis for our register-based surveillance. And we established the tradition of doing clinical trials in Finland, both phase 3 and phase 4, which we're right now doing a lot register-based, using the individual person identifier as linkage. And I'm very grateful to Helen Mäkelä and Juhani Eskola that they saw through this and thought big, that not just this intervention, but actually the many interventions to come. Over to you, Franziska.
Thank you, Hanna.